Welcome, everybody, again to another episode of the ideaXme show, the show that profiles the humans behind the really big ideas that are shaping the world and inspiring future creators. I'm Ira Pastor. I'm your health, aging, and longevity ambassador along for this ride. Here we are, 2019. And although we spent a lot of time on this show in the past talking about the chronic degenerative diseases responsible for human suffering and death, we can't lose sight of the fact that infectious diseases remain a leading cause of death worldwide, particularly in low-income countries, especially amongst young children. Three infectious diseases are still ranked in the top 10 causes of death worldwide by the World Health Organization, including respiratory infections, diarrheal diseases, and tuberculosis, which kill over 5 million people each year, an additional million or so with malaria. AIDS, still about 1 million deaths a year. A large amount of this is mentioned, low-income countries, and it's just still, infectious disease is something that we cannot overlook even in 2019. The antibiotic, by definition, is a type of antimicrobial substance that is specifically active against bacteria, and has been an extremely important type of antibacterial agent for fighting bacterial infections, and is widely used in both treatment and prevention nowadays. The modern era of antibiotics started with the discovery of penicillin by Sir Alexander Fleming back in 1928, and since then, antibiotics have truly transformed modern medicine. First prescribed in the 1940s during World War II to control bacterial infections. However, shortly thereafter, we began to see a trend that resistance, specifically the penicillin at the time, became a substantial, clinical issue, such that by the 1950s, many of the advances that we saw were somewhat threatened. In response to this, we developed new classes of antibiotics, the tetracyclines, erythromycins, aminoglycosides, and a wide range of, beta-lactam antibiotics, penicillin derivatives, cephalosporins, monobactams, and so forth. And these were discovered and then deployed into the clinic. Unfortunately, bacterial resistance eventually developed to all antibiotics that have been developed, including methicillin, sort of the bad boy on the penicillin ladder, with methicillin-resistant staphylococcus being identified in 1962, and also vancomycin, which is a true antibiotic of last resort, which was introduced in clinical practice back in 1972, but resistance showed up in 1979. And tangential to all of this, the big problem, simultaneously, while we see this antibiotic resistance popping up, the antibiotic pipelines of big pharma also simultaneously began to dry up as a result. And here we are in 2019, many decades after the first patients were treated with antibiotics, having a major problem on our hands. So the big question becomes, on this planet where we have an endless array of pathogenic bacteria that have survived for billions of years in quite inhospitable environments, and which over the course of only 100 years have sort of shrugged off everything that modern medicine, in terms of the antibiotic portfolio, has thrown at it, where do we look for future solutions to this problem? Well, some very creative thought leaders, including one we have today, believe the answer exists in another area of really tough organisms that live on this planet, namely the bacteriophages, which are types of viruses that infect and replicate within bacteria. Bacteriophages are one of the most common and diverse entities in our biosphere. Estimated there's something like 10 to the 31st power of bacteriophages living on the planet right now, more than any other organism on Earth combined, including bacteria. Interestingly, phages have been deployed for the last about 100 years or so as alternatives to antibiotics in certain countries like the former Soviet Union, Central Europe, and so forth, and they've been seen as possible therapeutic interventions against multi-drug resistant strains of many bacteria, and also have been shown to interfere with biofilm production and quorum sensing in pneumonia and cystic fibrosis and other diseases. So for today's guest, who I'm extremely excited about, it's going to take us further along this theme and sort of where the future is taking us, is Dr. Steffanie Strathdee. She is Associate Dean of Global Health Sciences and Harold Simon Professor of the Department of Medicine at University of California, San Diego School of Medicine. She's also an adjunct professor at Johns Hopkins and Simon Fraser University, and co-directs UCSD's new Center for Innovative Phage Applications and Therapeutics, known as IPATH, Global Health Institute, and the International Core of UCSD Center for AIDS Research. Steffanie has a PhD in infectious disease epidemiology. She spent the last 2 decades focusing on HIV prevention in various marginalized populations, developing countries, and has published over 600 peer-reviewed publications, and has recently begun to move bacteriophage therapy into clinical translation. Currently, she leads a multidisciplinary team of research on HIV risk behaviors among drug users and sex workers in the Mexico-U.S. border. 2009, she and her team were awarded a leadership award in international collaboration from NIDA and the National Institute of Drug Abuse, which also granted her a merit award for her research in Tijuana. In total, she's been awarded over $64 million in federal grants as principal investigator, and most recently has co-authored a book entitled The Perfect Predator, an epidemiologist's journey to save her husband from a deadly superbug, published in 2019, which discusses her experience in proposing experimental bacteriophage therapy to save her husband from a life-threatening multi-drug resistant bacterial infection. All that being said, Steffanie, thank you so much for coming on the show today.
It's a pleasure. Thanks very much for having me, Ira. Where would
Most
you like me to
definitely.
begin?
Basically, from the beginning, we usually give our guests the floor just to hear more about you, sort of where you grew up, your background, how you got interested in science and epidemiology, infectious diseases, and sort of the path that led you to what is, technically an extremely bleeding edge area of pharmacotherapeutic intervention.
Right. So as you mentioned, I'm an infectious disease epidemiologist. I was trained in Canada. I did an undergraduate degree at the University of Toronto in microbiology, followed by epidemiology. And
Mhm.
the HIV epidemic hit while I was still an undergraduate student. So I was poised to, seize upon this. And my PhD advisor and my best friend died of AIDS within a year of one another. So I've spent a lot of time, on a personal mission, really, to try to focus on this. But life really, threw a curveball at me in 2015. My husband and I, Tom Patterson, were both professors at the University of California, San Diego, and we were on vacation in Egypt. And we're having a great time. It was the last night of our trip. We were on a cruise ship that parked at Luxor. We were supposed to see the Valley of the Kings the very next day. And we were the only ones on the ship because there had been a terrorist attack in Sharm el Sheikh a couple of weeks earlier. And so everybody else canceled but us. And we thought that it was actually the best time to go because there'd be hardly anybody there.
Mhm. Sure.
Well, the terror actually came from within,
Mm.
not from without. And Tom got exceedingly ill after um, a meal on the cruise ship. And I just thought it was food poisoning because it was seafood, and we're traveling in a country that has some limited resources, and so and the Nile isn't the closest and healthiest body of water, so. We were concerned that he was going to have some problems, and I called the doctor to the ship, and he took one look at Tom and said, "Well, he's going through what's probably food poisoning. I'll just give him some antibiotics and some fluids, and he should be right as rain in a couple hours." He wasn't. He actually got worse. And I was going through in my mind, calculating the various incubation periods for different kinds of organisms that he might have acquired. But then he started complaining of back pain. And, although I'm not a medical doctor, I have a PhD, not an MD. I know just enough to get myself into trouble when it comes to medicine. But I knew back pain wasn't, obviously a symptom associated with food poisoning. So this is the middle of the night. I Googled the symptoms because I didn't have anything else at my disposal. And it suggested that pancreatitis could be, the diagnosis. Now, obviously, you don't use Google as your doctor. I called home. The head of infectious diseases here at UC San Diego is a colleague and friend of ours. And when I described Tom's symptoms, he said, get him to the closest hospital, go to the one that the expats go to. And there was no hospital in Luxor. There was only a clinic. The doctor took us to the nearest one. And there they did diagnose pancreatitis, which is an inflammation of the pancreas.
Hmm.
But that was the first of our, problems, but it wasn't the last because that was just a symptom of a much larger issue.
Sure.
We didn't figure out until he was medevaced to Germany that he had a gallstone that was blocking his bile duct, and it caused a giant abscess or pseudocyst to form
Okay.
in his abdomen. It was the size of a small football. And that created this perfect apartment for this superbug to move into. Later, the organism was sequenced and it was found to be an Egyptian strain.
Hmm.
We'll never know where he acquired it, but it's one of the nastiest. In fact, the doctor called it the worst bacteria on the planet. And its name is Acinetobacter baumannii,
Mm-hmm.
and it's an organism that I used to plate on my Petri plates back in the 1980s at the University of Toronto when I was a microbiology student. And then we didn't need anything more than gloves and a lab coat to deal with it. It was
Right.
considered to be a pretty wimpy organism. But over the last several decades, it's become something of a bacterial kleptomaniac. It acquires antimicrobial resistance genes from other organisms very easily. And so as we're throwing antibiotics at, whatever, pathogen we were waiting to get, cultured, it was killing off all the friendly bacteria in Tom's microbiome, and it left this superbug, which was resistant to the antibiotics that he was being given. So
Mm-hmm.
by the time they cultured this and this Acinetobacter baumannii, they were alarmed because it's associated with a lot of multi-drug resistance, especially from the Middle East. So
Okay.
we were in an ICU under infection control procedures that got even more strict. And then they did what's called an antibiogram, which is essentially the antibiotic susceptibility profile of the organism. That took a couple of extra days. And that's one of the issues. We don't have these kinds of point of care diagnostics readily available. So by the time we got those results back, Tom was probably sick for about a week. And the results were shocking. This was a highly drug resistant organism. It was resistant to 15 different antibiotics right off the bat. And it was only partially sensitive to three, and those were the carbapenems and
Mm-hmm.
colistin.
Mm.
So those are antibiotics that are only available intravenously. So they put him on those, a combination of those right away. But by the time he was medevaced back home to San Diego, it was resistant even to those. So it
Mm.
acquired new antimicrobial resistance genes in those few weeks, despite infection control procedures.
Amazing.
And so, incidentally, right around the time Tom got sick, there was a paper that was published in the top medical journal, The Lancet, and it was about the MCR1 antimicrobial resistance gene that was first discovered in pigs in China. And this confers resistance to colistin, and
Hmm.
that's our last resort antibiotic that was developed in World War II. It's not exactly a
Yeah.
wonder drug, but it's one that is highly toxic, but it can be effective in certain circumstances. And so that was a big concern when this paper was published. But by the time people started looking for the evidence of this MCR1 resistance gene, it was found that it was in 30 countries already,
Oh my
and
God.
Tom's organism had it. And so that was a shock because nobody was expecting that it was going to be so widespread. And it just points to the lack of our global surveillance for antimicrobial resistance, which is a huge problem. Because if you don't know what you're dealing with, these organisms are causing infections that are undetected, they're
Mm-hmm.
undiagnosed, and then they become essentially untreatable.
Mm.
So Tom, was really, becoming the poster child of this post-antibiotic era that people have been warning us was coming. And
Sure.
as an infectious disease epidemiologist, I knew that superbugs were a problem, but it wasn't really till it hit me, in my own personal life. And when I realized that this was an organism that I dealt with as an undergrad, and now it's like the number one public enemy on the World Health Organization's list of the 12 most dangerous superbugs to human health. So that was the beginning. So I'll just pause there to see if you wanted me to clarify any points before this terrible story continues.
No, no, I appreciate it. I mean, it's not a great story. It's an extremely interesting story, and I have to say the same thing. When I looked at the paper, and I'm it had I haven't I haven't had microbiology for for 30 years, but I don't remember it. I remember there was this group of stuff. I will never need to know about those ones, but yeah, it's just wow. I mean, the fact that it. Uh, of all things, such a rare, the ones that we don't look at, there's that top list of whatever they're called of,
The escape pathogens.
right, the escape
Yes.
pathogens, and here you have, it's bizarre, but please continue. I don't want to get in the way. I definitely want to hear. more of
Yeah,
this.
but since you brought that up, just for your listeners,
Please,
the
please.
escape pathogens, I mentioned this in our book, The Perfect Predator. One chapter is actually called No Escape. And escape in this way is spelled ESKAPE.
Mm-hmm.
And each one of those, letters stands for a different superbug. So the A is Acinetobacter baumannii, the one that Tom had. And so those are considered to be some of the most critical, superbugs that we're dealing with around the world. But even since that list was, generated several years ago, it's become much longer now because so many more organisms have acquired these, resistance genes because they're swapped back and forth between different bacteria. And
Mm-hmm.
so basically, Tom got sick in November of 2015. He was medevaced back to San Diego just before Christmas. And at that point, they repeated the antibiogram, and then they saw that the antibiotic susceptibility showed no susceptibility to any existing antibiotic. And
Okay.
so they tried to look at different combinations of antibiotics to see if any of those would be, potentially useful. And those we struck out there too. So the doctors had really, this terrible choice. Either they could operate on Tom and try to take this abscess out of his abdomen, but then risk the possibility of septic shock.
Right.
If that organism got into his bloodstream, there was no antibiotic to cover it, and he would likely die. He was very weak at this point and had lost, close to 100 pounds. And the other choice was to put catheters or these drains in the abscess in his abdomen to try to drain off the infected fluid with the hopes that it would shrink and that his immune system would get stronger. And so that's the route that was advised. There was some debate over which would be the best route to go. There were none of these were great options.
Mm-hmm.
So for a while, his own immune system was hanging in there. This cyst was shrinking a little bit, but the infection kept spreading to new collections. So the problem of source control, as the physicians described it, was an issue because all of this fluid that is draining out of this abscess is, debris from the bacteria and cells that are dying, and so
Right.
these drains get clogged, and so it's kind of like, a carburetor and, like a car
Right.
that's like, has this oil, but then, there's all this crap that kind of gets caught in it, and it causes a clogging up. So that's essentially what was happening. And this one day, Tom sat up in bed, and one of the internal drains inside him slipped, unbeknownst to us at the time. And it poured all that infected fluid into his abdomen and into his bloodstream, and immediately he went into septic shock.
Mm.
So at this point, I should tell your readers what that looks like, because people hear of sepsis and septic shock, but when you see it happening, you don't necessarily clue in. And again, because I'm not a physician, it was something that I had to learn. And Tom
Sure.
had 7 cases of septic shock within the course of his illness. So I got really familiar, unfortunately. So the first thing that happens often is there's a dramatic drop in blood pressure and an increase in temperature and respiration rate and heart rate. And it happens very quickly. And the patient is often either perspiring or might be, urinating profusely all of a sudden. And often they have a clammy appearance. And then something called rigors occurs, and that's when the shakes.
Sure.
And these aren't just like when you have a fever and you get like a little bit of a chill. These shakes are so bad that like the bed frame hits the wall. And that's what was happening to Tom. So within the span of about 2 minutes, he went from saying, "I'm cold, I'm not feeling very well," to, and he just tanked. And luckily, a doctor and a nurse were there at the time. They got him rushed back to the ICU. It wasn't until they did a CT later that they realized that this drain had slipped, but we knew that he was having a septic shock episode.
Sure.
Unfortunately, they had to put him on a ventilator, so that's like or a respirator, as some people call it. That's a form of life support to help you breathe when your
Sure.
lungs are failing. And He was put into a medically induced coma to give his body a rest for a couple of days. And of course, he wasn't going anywhere for a while. I mean, we had thought that he was about to be discharged to go to a long-term acute care facility before this happened. And when this happened, it just, it turned everything upside down. And so once they did the CT and they realized that basically this drain had slipped, they realized his body is now fully colonized with this organism. It's no longer just contained in this pseudocyst. It's the worst thing that could have happened because now it's everywhere. How are we going to contain it? There's no antibiotic left. And so from that day onward, and that was, I think, January 19th of 2016, he was dying a little bit each day.
Right.
And he, had moments of consciousness after he was brought out of the medically induced coma, but he had long periods where he was comatose and having these crazy hallucinations that we detail in the book because he remembers them all vividly, which is very unusual. And so at this one point, the doctors really were turning to me and saying, it doesn't look like he's going to get better. And by the time I realized that he was really dying, they asked me if I wanted, to put him on kidney dialysis because his kidneys were starting to fail. His heart was already failing. And I decided to ask Tom what he wanted to do. Even though he was, pretty much in a coma, I thought, I've heard that people can still hear sometimes.
Sure.
And I shouldn't make this as a unilateral decision. I should try to find out if he wants to live because, I'm trying to decide for him because I had power of attorney. So I had this conversation with him where it was one of the hardest I've ever had in my life, where I asked him if he wanted to live. And I told him that I loved him and wanted to grow old with him, but if he wanted to live, I needed a sign, and that he should squeeze my hand, and that if he wanted to go, I'd understand. And, I waited about a minute and he squeezed my hand really hard. Much later, he told me what was going on through his mind when, because he did hear me and he did squeeze. And I'll tell you about that later because, this moment was obviously pivotal because I told the doctors, if you have to put him on kidney dialysis, do it because I'm going to try to research this on my own.
Right.
So I went home and because I'm a researcher, I know how to do a literature search. So I turned to PubMed, which is the search engine that the National Library of Medicine makes freely available to anybody. It's like, Google Scholar on steroids, and it has all the published information that's peer-reviewed in top journals that's available. So I put in keywords like antimicrobial resistance, Acinetobacter baumannii, alternative treatments, and up popped a paper that had, alternative treatments that were not widely available, and that, some, one was a vaccine. If, maybe, years in the future that might be available, but it certainly wasn't an option. And one of the was phage therapy. And I thought, I know what phages are. You call them phages, but the discoverer of bacteriophage was a French Canadian. So phage and phage are used
Right.
interchangeably. Bacteriophage were found in 1917 by French Canadian Félix Durel, who deduced that he was witnessing some agent that was taken from the feces of people who'd survived from dysentery that was killing, the same organism in patients who were dying of dysentery. And he put the fecal matter through a Pasteur filter that screens out not just the fecal matter, but it screens out bacteria.
Right.
And the liquid that he, filtered through it was still able to kill this organism. So he deduced it had to be smaller than a bacteria, and therefore it must be a virus that preys on bacteria. And he called it bacteriophage after the Greek phagine, if I'm pronouncing it right, which is to eat. So it means the eater of bacteria or bacteria eater, bacteriophage. So, and as you told the listeners in the beginning of the podcast, it wasn't until 1928 that penicillin was discovered. So phage was actually discovered prior to penicillin.
Sure.
And of course, nobody could see these phages. They were smaller than bacteria, so they couldn't be seen with a light microscope. And the electron microscope wasn't developed until 1940 in Germany. And so we didn't have, the ability to share information across the countries and the oceans like we do now. So nobody knew what phages really were. Felix was adamant that they were viruses. Other people thought that maybe they were enzymes or a kind of protein. And there was a fierce debate about this. But nevertheless, Felix actually experimented on himself and some of his lab mates and his family before a couple of years later, in 1919, he used a suspension of phage to treat children with dysentery in Paris during an outbreak. And it cured them within 24 to 48 hours. And it was, really miraculous.
Amazing.
So from there, he went on to use phage to treat both animals and people. And he was very famous for this period of time. He was actually the inspiration for the book Aerosmith that Sinclair Lewis wrote, that won the the Pulitzer Prize. And so at that point, there was efforts to commercialize phage products, and they didn't really know much about phage because the scientific method wasn't clearly established. There was no knowledge even that DNA had a triple helix, anything like that. We didn't know, for example, that phage have to be matched to specific bacteria. It isn't like there's a universal phage that kills all bacteria.
Right.
They have to match, almost like a lock and a key. So what happens, and we know now what phage look like, they come in all shapes and sizes, but the one that people recognize the most looks almost like a little alien spider. And they've got a capsid, which is its head, which is usually in the shape of an icosahedron, which looks like Disney's Epcot Center, but in miniature. They've got often a long neck, and then these spindly little fibers, and those attach to a receptor on a bacterial cell. And if it's a match for them, then it locks onto that bacteria, and it shoots its genetic material from its capsid through the hollow tube of its neck into the bacterial cell. And if it's the kind that we want, which it undergoes what's called the lytic cycle, I describe it in our book as phage rage, where it takes over the bacterial cell genetic machinery and it turns it into a phage manufacturing plant. And so all of these baby phages are made, they're called virions. And when given the kill signal, they burst out of the bacterial cell, killing it. That's called lysis. And then there's all of these baby, phages that can go on to kill and infect new bacteria. As long as there's bacteria that match to it, they'll keep multiplying. But then after there's no bacteria left, they are excreted by the body naturally and through the reticuloendothelial system, which fancy name for what the liver and the spleen are part of. So they basically filter out the rest of the phages, and there's no harm to the rest of the bacteria in the microbiome. So this self-limiting property is actually very attractive because with antibiotics, which are generally broad spectrum, they kill not just the pathogenic bacteria that we want to kill, they kill the friendly bacteria in our microbiome as well. So phages, of course, we didn't know a lot about this at the time, have got this wonderful kind of natural living antibiotic kind of approach to them, that if you can find the right phage that matches the bacteria you want to kill, it acts as its own kind of living antibiotic. So now the question was, could we find phages to match Tom's bacteria? And that's the idea that I had. That was my main contribution to this whole story, but it was an important one. I posed it to the head of infectious diseases, Dr. Chip Schooley, the same guy that I called when Tom was sick in Egypt. And he said, wow, what an interesting and intriguing idea. If you can find phages that match Tom's bacterial isolate, I'll call the FDA and get approval for compassionate use because this is experimental treatment. It's not available in the US.
Sure.
It's not licensed, and it's only being used in the former Soviet Union. Now, that is a digression that I'll just spend a little bit of time on. We get into that medical history in our book. So why the Soviet Union and why isn't it available here? Well, it goes back to, the 1920s and 30s, because when penicillin was discovered in 1928, it took some time for them to figure out how to isolate and expand and manufacture penicillin from the mold. And the pressure of World War II made that happen. So by
Right.
1942, the first US patient was treated with penicillin. And then, it was, really seen as a war secret, but that one that was going to be very important to save the troops' lives, many of whom were dying from gangrene and diphtheria and all sorts of other preventable infections. And so penicillin was a wonder drug, and phages were these things that were finicky. They had to match to the specific bacteria, and they didn't always work because nobody knew that you shouldn't be, sterilizing the preparations or you'd kill the phages and things like
Mm-hmm.
that.
Mm-hmm.
So the West thought phage is just too busy and finicky, and we're going to forget about it.
Right.
And penicillin wasn't widely available in some parts of the world. So the former Soviet Union, okay, was using phage. In fact, Rommel's troops in World War II were found to have phage preparations in their satchels.
Mm.
So it was widely used in Japan, Russia, and in Germany, and it's been used in the former Soviet Union ever since. In Georgia, the Republic of Georgia, that is, there
Mm-hmm.
are two phage therapy centers. There's one also in Wrocław, Poland, and they see it as standard of care. But
Sure.
in the US, because it was taken up so vigorously by what was then an enemy during World War II, if you were into phage therapy, you were labeled a pinko commie. So Felix Durrell's biographer, Bill Summers from Yale University, has written extensively on this topic and has shown that there was a real geopolitical bias that put a cloud over phage therapy, because in the 1950s and 60s, most of the Nobel laureates in science were winning their prizes for work that they were doing on phage, but they were using phage as a tool to discover the whole fields of molecular engineering and, recombinant DNA and, cancer biology and even CRISPR and
Right.
gene display or phage display. Sorry, those all were incredible discoveries that it wouldn't have been possible without phage. But the whole phage therapy approach, the therapeutic potential of phage, was relegated to the back burner. So that was a digression, but it's important to know that it's seen as the standard of care there, but not in the West.
Let's just talk about break off for 1 minute and talk about that a little further, because I think it's an important link. Obviously, after your story to the big question, and how this goes to hundreds of millions of people ultimately, because when I read, I find it a fascinating topic. I spent most of my time the last 35 years or so in your traditional pharmaceutical industry, and I know how most of the people inside big pharma think of, if it's not a little white pill, it's not a drug, and
Yeah.
everything else is weird. But when I read about phage, and sort of the issues with regard to strain specificity, as you mentioned, and multiple phages may be needed for, and so forth and so on. I understand the way the regulators of yesteryear think, and the way pharma continues to have the reductionist view. However, that being said, here we are in 2019, and more than ever before, there is a lot of what we'll call, I'll throw into the basket of weird or undrug like stuff getting into the clinic. So, I mean, we have, we have microbiome cocktails. We have living organisms like leeches and maggots, which are FDA approved now as medical devices for non-healing wounds. We have botanical drugs where the FDA for the first time has said, okay, you might not know everything in your little gamish of stuff, but if you can standardize it in some fashion. And I think this is very important with regard to phage because there is now potentially, unlike in the past, this opportunity to take something that is, you open it up, it doesn't look like your traditional drug, but it's doing amazing things, and we need to find the path to move it forward to not just one patient, but hundreds of millions ultimately, because we're at a really bad point. So I'd love if you could talk a little bit about that and then ultimately link it into sort of some of the experience with, your associates contacting FDA, the experimental authorization, and so forth, and the N of one case, and then your thoughts to how that was received
Right.
by FDA, and how you feel about this potentially now in 2019 being able to finally make it to the big show for everybody else.
Right. Well, I mean, I can't speak too much for the FDA, but obviously they've been grappling with, issues like you spoke of with fecal transplants, for example,
Sure.
where, you know, you're transplanting organisms from one person into another, and those are very difficult to standardize. So, the regulatory pathway was one of the hurdles that was preventing phage therapy from being,
Mhm.
expanded in the West. But so was the lack of rigorous controlled clinical trials.
Sure.
So in the former Soviet Union, they don't have the ability to purify their phage preparations by removing the endotoxins, which are essentially the bacterial debris that occurs when you're manufacturing the phage preparations at a large scale. And
Okay.
so they have just been using phage topically, and they haven't felt the need or the pressure to do a clinical trial because it's already standard of care there. But
Mm.
in the US, we don't have the data that's necessary to convince the FDA to license it. So those clinical trials need to get done. They're getting started
Absolutely.
now. But certainly at the time when I proposed this idea, to my colleagues, most of them had never heard of phage therapy. Others thought, okay, this is like, one of them called it a Hail Mary pass at the last minute of the game where the quarterback is blindfolded, and you're throwing this ball 100 yards down the field, but everybody realized that he was going to die.
Hmm.
He was going to die real soon. And so that's what the FDA said to my colleague Chip Schooley. They said, this is the kind of case that we've been waiting for. We know all about phage therapy, and we want to make it, available if we have the data, but we don't have any data. So if, here's a patient who's dying, his family members are agreeing to do it. We've got a doctor and a medical institution who's willing to cut through the red tape to make it happen. And, we've got a phage community that's stepping up to the plate, because I wrote total strangers and asked them to help. And Texas A&M University and the Navy Medical Research Center in Frederick, Maryland, who were all
Hmm.
total strangers to me, said that they would turn their labs into command centers to see if they could find phages to match Tom's case. So that was what they did. And it took a couple of weeks to get the phages purified once they found matches. And then we had to, remove as much endotoxin as possible, because Tom was fully colonized, and we realized that we wanted to treat him intravenously, because if there was a hidden reservoir of bacteria that the phage couldn't
Hmm.
reach, it was thought that the bacteria could become resistant very quickly,
Sure.
because the bacteria are multiplying every 20 minutes. The phage are multiplying even faster, but this
Okay.
is like evolution on a rapid miniature scale. And so um, we luckily got the phage in time and injected them into Tom's bloodstream. They were a billion phages per dose. And that was the scariest day of my life doing this, because I knew that if we didn't have the endotoxin level low enough that we could kill him, right? And now the people who have had a lot of clinical experience with this in the former Soviet Union would say, oh, well, those fears were, not, warranted, but they typically don't inject phage into the bloodstream there because they don't know what endotoxin level they have because they don't have the resources to measure it. So we were terrified, but Tom basically, not only did he stabilize, but 3 days after we started injecting the phages into him, he lifted his head off the pillow and kissed his daughter's hand. And everybody in the ICU freaked out. And of course, he had a long recovery. He'd lost over 100 pounds by this point. He lost all his muscle mass. He had to learn how to, swallow again. He had to, be weaned off the ventilator. He had to learn how to stand, walk, everything, and he didn't get out of the hospital until August of 2016. But if he'd had phage therapy, we're convinced very early on in the course of his illness, he would have been able to walk out of the hospital.
Absolutely.
And now, since Tom's case, we've treated 6 other cases at UC San Diego, dozens of others we have consulted on internationally, not the least of which was the first genetically modified phage to treat a girl with a systemic superbug infection in the UK. That was published in May of this year, 2019. And it's remarkable for 2 reasons. First, it was the first time a genetically modified phage has been used successfully in a human being. And second, it's a Mycobacterium abscessus infection. So that's a non-tuberculous mycobacteria.
Okay.
But mycobacteria that's the same genus as tuberculosis. So her infection was Mycobacterium abscessus. Mycobacterium tuberculosis causes TB, which is the biggest bacterial killer in the world. So it lends some hope that one day TB might be able to be treated with phage therapy once appropriate studies get done.
Well, I don't mean to interrupt you. Why do you have did you have to CRISPR why did you have to modify that phage?
Okay, so what I skipped over earlier is that there's 2 life cycles of a phage.
Okay.
And the first is the lytic cycle that I described, and I called it phage rage, right?
Sure.
And that's where like lysis happens and the bacteria is taken over by the phage that turns it into a phage manufacturing plant. That's what we want for therapeutic applications of phage. The other life cycle of a phage is called temperate phages. And they undergo what's called lysogeny. So after they attach with their receptor and their genetic material enters the bacterial cell, for reasons that we don't understand very well yet, their genetic material gets integrated into the bacterial cell DNA and becomes a prophage. And it sleeps there. I call it hitting the snooze button is how I describe it in the book. And we don't want that for a number of reasons. So this life cycle is called the lysogenic life cycle. Temperate phages do this. And we don't want this because, first, the bacterial cell isn't killed. Second, some of these phage can actually emit a signal that makes that bacterial cell resistant to attack by other phages, including lytic phage. So it kind of, prevent it. So it's kind of like what my colleague Dr. Graham Hatfield calls colludes. The phage colludes with the bacterial cell to help it because it's in both of their best interests so that they don't get killed, right? The third thing that can happen is that these phage that integrate into the bacterial cell DNA can carry antimicrobial resistance genes or toxin genes. So that could cause harm. So for reasons that we don't really know, some bacteria, the phage that attacks them tend to be temperate. They tend to be the lysogenic kind. So phage that attack C. difficile, which is C. diff, as people will know it, or the phage that attack Borrelia species, which cause Lyme disease. Those phage are almost always temperate phage. So in the case of mycobacteria, its temperate phage predominates. And so you want to be able to convert these temperate phage to lytic phage. So what my colleague Graham Hatfield did was go in and clip the repressor gene. He didn't use CRISPR, although CRISPR-Cas technology can be used to gene edit phage and is being used by other groups. But he was using this approach called recombinering, and
Okay.
it just clipped out the gene that makes these phage undergo lysogeny. So it forced it to become lytic.
Okay.
And because those are the only phage that he could find. There was one lytic phage and I believe two lysogenic phage. So he was able to make a phage cocktail of this.
I
And
see.
the UK government gave approval because essentially they didn't consider it to be genetically modified because you weren't adding a gene. You were
I
just
see.
taking away a gene. And theoretically that phage could have lost that gene on its own and we just helped it along.
I see.
So I wasn't involved in this case directly. My colleague Chip Schooley advised on the clinical application, and it was based on my husband Tom's treatment. And so it's clear to say, though, that without Tom's case, this girl, her name is Isabel, would have died. And
Hmm.
we're in touch on Facebook, and I'm also in touch with her mom. And, she's written me and said, when are you coming to visit? I'll make cupcakes. I mean, this is coming from a girl who was in hospice because she had cystic fibrosis. She'd had a double lung transplant and she was immunosuppressed. So these kinds of infections often, are a problem for people with CF. And she was able to leave the hospital within 10 days. And she's now taking her A levels, learning to drive, has a part-time job. She still has this infection. She's still getting phage therapy. It's a very slow-growing organism. So unlike Tom, who was able to clear his infection with the help of phage within 3 months, Isabel still has hers, but it's the phage therapy has gotten it under control so that she can live essentially, a much healthier life, and she wouldn't
Absolutely.
have made it otherwise.
Fascinating. Fascinating. Um, you know, tangentially to this, 'cause you mentioned the sepsis component in Tom's case. There was a paper, I have it somewhere here. I think it was maybe from 2018 or something, but it was specifically focusing on the immunomodulating properties potentially in phages as well, and having sort of this double whammy potential. Have you studied all about some of? I know it's early days still, but potentially because I know once again going back to pharma, sepsis is one of these things where pharma bats 0 and sepsis, I don't know how many clinical trials there's been, but sepsis bats 100 or something. Have you begun to look at any of the immunomodulating potential properties of phages alongside their lytic potential?
Well, yeah, I mean, just to clarify, so I'm not a virologist, so my role with iPath and moving things forward is really to raise awareness and
Hmm.
to, kind of convert the scientific lingo into something that people can understand. So my understanding of there is a hidden benefit of phage that when they lyse bacteria, when they kill it,
Hmm.
that bits of phage are attached to bits of bacteria,
Okay.
cell wall, like for example. And so that can act as a primer of the immune system's response. So it acts kind of almost like a vaccine would, and it can prime the immune response. So there are people that are studying that aspect of it. But
Uh-huh.
I think more exciting is this whole potential now of potentially genetically modifying phage,
Okay.
so that we can make them have a broader host range. So in my husband's case, we had to not just match the genus and the species, but the phage had to be matched to Tom's bacterial isolate.
Sure.
That's because the host range for Acinetobacter baumannii phages is extremely narrow. So we were very lucky to find the phages to make a cocktail. And I should say we had a phage cocktail, and that's what we, advise people to do with phage therapy so that you're not treating a single phage. Because imagine the bacteria is being attacked by, a variety of different things. And if it's being attacked by more than one phage at the same time, especially if they're phage that attack a different receptor, it's harder for the bacteria to mutate to evade them all.
Sure.
So if you're only using one phage, the resistance can happen very quickly,
Right.
depending on how quickly the bacteria multiplies.
Sure.
So
Okay.
in Tom's case, if we want to get a handle on Acinetobacter baumannii, the bacterial infection Tom had, it may be necessary to genetically modify phages so they have a broader host range. So those are the kinds of studies that are ongoing now. There's also some evidence in Tom's case and others that phage can be synergistic with antibiotics. So even if phage was to go through clinical trials and not be superior to antibiotics, I kind of doubt that, but we
Right.
need to do those studies before
Sure.
we can license phage in the West. But if its sole purpose is to resurrect a failing antibiotic regimen because of the synergy that, it's putting, pressure on the bacteria from two sources, the phage and the antibiotic, and the bacteria die. Those that live that have some kind of mutation are often susceptible to the antibiotic. So that would be a game changer because, it means that antibiotics that have been sitting on the shelf because they don't work anymore could be useful again. So pharma is finally paying attention to phage in terms of its therapeutic potential now.
Hmm.
A number of companies like Johnson and Johnson are investing heavily in this area, and clinical trials are necessary. And we formed the first phage therapy center in North America, IPATH, at UC San Diego, with some seed funding from our chancellor. We're fundraising, so if anybody wants to help out in that way, obviously. But more importantly, along the way, while we're getting ready for clinical trials, we're receiving and helping patients like Tom who are desperate to help their loved ones, or physicians whose patients have run out of antibiotic options, and we're phage hunting for them. Now, we've been doing this for a couple of years now, and the biggest barrier is finding the phage.
Hmm.
Because as you mentioned in your opening remarks, there's 10 to the power of 31 phages on the planet. That's just an estimate, but that means 10 million trillion phages. So that's both an advantage and a disadvantage. It's an advantage because it means that there's a limitless supply of phages that you could make a cocktail for to attack virtually any kind of bacterial infection
Hmm.
you want to treat. But you have to find them, right?
Right.
And they're like, people have different like repositories of phage all over the world, and some have been sequenced, some haven't, some are lysogenic, some are lytic. And so what we want to do at IPATH is actually develop a ever expanding phage library to match an ever
Sure.
expanding library of superbugs, because they're co-evolving, right? They've been co-evolving for like 3 billion years, and
Right.
they're going to continue to do that. But if we have all these phage that are kind of essentially in the equivalent of a walk-in freezer, and they're
Sure.
characterized with that means we know their genetic sequences, we know which receptors they hit, we know which phages work well in combination with one another, then we can actually respond to outbreaks, emergency cases, and have those phage available for clinical trials and other studies. So that's what we're working on right now, and we would make this open source available worldwide.
What just off the top of my head, what does phage hunting look like? I mean, is it sort of like old school natural product screening where you would just go look at soil samples and how do you hunt phages anyway? Just
Well,
what's
there
involved?
is a real crude aspect to this, and in some ways, the whole approach hasn't changed in 100 years. We do have, things like high throughputs, approaches, and next generation sequencing and
Uh-huh.
things like that. But the phage hunt itself, you need environmental sources, unless you're
Okay.
going to, generate a synthetic phage from scratch. There are
Right.
people doing that with metagenomic techniques, like,
Sure.
piecing together like fragments of DNA, for
Hmm.
example. But, an old-fashioned phage hunt is you go to wherever there's a lot of bacteria. So a sewage treatment facility, or a
Hmm.
pond, or, you know, um, soil, and you get that sample, and you add water to it, and
Hmm.
you can put a drop of that water on to a Petri dish that includes the bacterial culture that you've streaked and grown on a Petri dish
Okay.
ahead of time. And then you incubate it for 24 to 48 hours. And if there are phage that match that bacteria, you'll see a hole in the agar. So even though you can't see the phage with your naked eye,
Sure.
the evidence that the phage is there is like essentially a hole in the agar. It looks like Swiss cheese. So then you know that you've got phage that's active against that bacteria. You can pluck it out, add more bacteria in a suspension, grow it up, and then you've got to, purify it. And the purification takes longer. And in Tom's case, that was the longest part of the whole process. And so you need, if you're going to prepare phage for clinical application in a non-emergency, you need to have GMP facilities, like,
Sure.
good manufacturing, procedures in a facility that's high grade, that can be, regulated. But in emergency cases, the FDA will give approval on a case-by-case basis with non-GMP phage.
It's completely a fascinating story, and I mean, I really appreciate you telling us, and anything we can do to increase the awareness of this, because it's, I love, aside from biotech, I love the history of this field. And when I see things, I mentioned earlier with the living microbes, and then some of the stuff that's happening. I used to work at GlaxoSmithKline, and they're getting involved in all this electroceutical business now. So all these ideas from 100 years ago, which were forgotten, including this. I like to see them showing up again in 2019 and changing the world, which is what you're
Yeah,
doing.
well,
And
that's my
it's,
handle on Twitter, is
yeah.
changing the world. And also, we wrote our book because we realized that phage therapy has had a lot of false starts. Most people aren't aware that, the superbug crisis is a more immediate threat in our lifetimes than climate change. By the year 2050, 10 million people a year are going to be dying from superbugs unless there's a dramatic change. And we don't see any new antibiotics in the pipeline that are going to turn this around. And
Yeah.
phage therapy is now considered to be the most important alternative to antibiotics that's out there. We don't see it as ever replacing antibiotics, although some people do. But I mean, my colleagues and I think that it will be an important adjunct.
Hmm.
But to get there, we need to have clinical trials. We need to have a phage repository that's standardized for other people to use, and that's what we're working on. So this has changed my life and my husband's life, but to know that it saved other people's lives, it makes us feel like maybe this is the reason we're on the planet.
There you go. There you go. Steffi, I just want to, moving from the science, we usually just go at the end of the show when we wrap up into sort of the science fiction. But I need to ask you a question about sort of people in the past that have inspired you, and if there's scientists, philosophers, artists, whomever, that sort of are not around anymore, but who you might have wanted to meet, that maybe set you on this path, whether it's Alexander Fleming, whether it's somebody like of that Shakespeare, whomever. Is there somebody that you would like to point out that if you had a magical time machine on hand, you could take a journey to meet them, you would like to do?
Well, it's interesting that you should ask that, because in our book, before every major section, I have a quote from a scientist
Hmm.
that really inspired me to help me kind of get through the ordeal that I was dealing with. So, for example, Sir Isaac Newton said, "No great discovery was ever made without a bold guess." And so
Absolutely.
one of our chapters is the bold guess, and that was injecting 1 billion phages into your husband's body to try to cure him from his superbug. I mean, it sounds crazy, but look at, what happened. Now intravenous phage therapy is being used, regularly. And there's been over 50 cases that we know about worldwide, and inspired by Tom's case. But I think on a more personal level, Barbara McClintock is
Sure.
somebody who was a real inspiration as well. And, she came up with the whole idea of jumping genes when she was studying corn. And she was kind of ridiculed for her science for a while. And she said, if you have a gut feeling that you're on the right track, don't let anybody deter you. Just keep at it. And so, even though other people thought that what I was doing was crazy, I knew that, if this worked, it was not only going to save Tom, but had the potential to save other people.
Hmm.
And so that's where it left off 3 years ago. But this field is still evolving, and I'm just really honored and proud to be part of it.
Well, it's an honor having you here, talking with us for a little while, and we will when the show goes live on all the different platforms, we will put links to you and iPath and your book, and try to get the message out as much as possible. Just really, once again, thank you for everything you do, not just today, but in building the future. For everybody watching and listening around the world, it's been Dr. Steffanie Strathdee, Associate Dean of Global Health Sciences, Harold Simon Professor of the Department of Medicine at UC San Diego School of Medicine, head of the Innovative Phage Application and Therapeutics Center, and also author of The Perfect Predator, Epidemiologist's Journey to Save Her Husband from a Deadly Superbug. Keep an eye on everything she's doing, because it truly is an extremely important time when it comes to this area of human health. And the big warnings that we've been seeing for years are coming to fruition. And it's scientists like Steffanie that are taking us the next way and protecting us and saving the millions that unfortunately would be killed by these superbugs if not for these newer technologies. So once again, Steffanie, thank you so very much for coming on the show, and thank you for everything that
It's a
you're
pleasure.
doing.
Thank you very much, Ira.
Definitely, most
Okay.
definitely.